I hope you're seeing the right screen. It's a pleasure to present to you some of our work on a technology we call pharmacoscopy, which is really nothing else than image-based high-throughput drug screening in primary human tissues. And the tissues we work with, the images that we generate in this approach are some examples are shown here. This is a fresh primary surgery piece of tissue from a patient suffering glioblastoma, so one of the most deadly cancers out there. In the middle, you see a bone marrow biopsy of a patient suffering multiple myeloma, and on the right, you see a field of view of healthy donor peripheral blood cells, like they're floating around in all of our veins. And obviously, we do immunofluorescence and high-throughput drug screening on exactly these kinds of tissues. So a quick disclosure, I was the scientific co-founder of Allsight in Vienna, which has been acquired by Exscientia, and I received research funding from Roche. So pharmacoscopy is the catch-all term that we gave this process of personal image-based drug screening. And we call it personal because the tissues go out of the person and directly onto the screening plates, or almost directly onto the screening plates, keeping the relevance of our phenotypes and drug screening results as close to the person, to the individual as possible. And that's one of the defining features of the workflow we've set up. In my lab, also in the company, and also in the previous lab at the Center for Molecular Medicine where I was, we have been running pharmacoscopy and adapting it to be compatible with many different kinds of tissues. So in our lab, for instance, we work with bone marrow, skin tissues, colorectal cancer tissues, obviously peripheral blood, lymph nodes, lung tissues, thyroid, and brain. So pretty much what we see is that this approach is compatible with any kind of human tissue you can imagine, which is of course very promising for its future potential. If it's a fluid biopsy, we purify out the nucleated cells. If it's a solid tissue, we dissociate, gently dissociate, it can even be a partial dissociation, and then distribute these nucleated cells or cell clumps into 3D4-well drug screening plates that have been previously prepared. And some of our past publications already showed that some of the applications that we can get out of this approach, including finding drugs that specifically kill cancer cells while leaving healthy cells alive, but also screening in high throughput or medium high throughput in primary tissues for immunomodulatory properties of drugs. For instance, scoring spatial interactions between effector immune cells, such as T cells, and a target population, for instance, cancer cells in a biopsy. The incubation is typically fairly short, 1 or 2 days, after which we fix and stain. We more often than not now perform multiplexed immunofluorescence with a single round multiplex immunofluorescence protocol we've developed. This picture here shows some 8 different stains at the same time. Then we perform high throughput automated confocal microscopy, single cell image analysis, deep learning, mostly using conventional convolutional neural networks, and then big data analysis to turn those images and data sets into meaningful insights. Just 2 days ago, we published the final analysis of what we think is one of the first successful prospective interventional clinical studies where patient treatment was being guided based on such drug screening results. And this was the EXALT-1 study, and it's now published at Cornell et al. In Cancer Discovery. And the interim analysis was published a few years ago in Lancet Hematology. And the process from a conceptual point of view couldn't be simpler. You get a blood biopsy of a patient suffering an aggressive blood cancer. These are patients that have exhausted standard of care, so the clinic doesn't have sort of approved treatment options for these patients anymore. We then immediately purify out nucleated cells, distribute them in 3D4-well based image-based screening plates. In this case, testing 139 different treatment options, all clinically approved anti-cancer drugs at different concentrations and replicates. And then after incubation, fixation, imaging, and staining and imaging, we just prioritize the drugs on how well they killed the cancer cells specific. So if a drug killed the healthy cells, left the cancer cells alive, obviously that's not something we want to replicate in the patient. Most of the drugs typically don't do anything. So there's no differential or specific killing of the cancer population. And the recommended treatments that we sent back to the clinicians as a recommendation for the treatment of that particular patient were those drugs where what we call the relative blast fraction, the percentage of cancer cells, was specifically reduced compared to the percentage of cancer cells seen in the control conditions. So this has an internal toxicity control. We do immunofluorescence matching the patient's pathology and diagnosis. And this was 5-channel automated fluorescence microscopy and bright field imaging for typically 2 3D4 plates per patient. And remarkably, we had a 5-day median turnaround time for this process. So it's highly automatable, it's very efficient, and it's faster than even just classical panel sequencing and things like that. A real world example, and this example has been floating around for a while now. This was one of the first patients that was treated based on pharmacoscopy recommendations. This was a patient before we received a sample of him with an aggressive diffuse large B-cell lymphoma. The patient was in palliative care, exhausted all standard treatment options, chemotherapy, immunotherapy. We received a lymph node biopsy, tested 104 treatment options ex vivo, calculated this relative blast fraction for B-cell positive, B-cell marker positive cells in that sample. And we found only 6 drugs that specifically and significantly reduced that fraction of cancer cells for that particular patient sample. The second best of which was the BTK inhibitor ibrutinib. The patient received that as a single agent treatment based on our recommendations and achieved a long-lasting complete remission. By now, and what we've just published, is that 56 blood cancer patients were treated based on our recommendations. And we see significant improvements in, for instance, the progression-free survival compared to the prior treatment response of the same patient cohort. So if we just compare how well did the patients do previously and how well did they do afterwards when we gave a treatment recommendation, we see a significant increase in progression-free survival. And there's this exceptional subset of patients that are achieving exceptional responses. So that means a response that is 3 times longer than what the clinicians would have expected for such patients. So that is highly encouraging. Another very encouraging signal that we saw out of this image-based screening approach for personalized medicine is that there was a cohort of patients that still had standard of care treatment options available to them. And we compared the overall survival of the patients that went on to pharmacoscopy-guided treatments versus standard of care. And actually, the overall survival was significantly improved for our pharmacoscopy-guided subcohort as well. So that makes this one of the first successful functional precision medicine approaches ever. Similar results are not available for instance, organoids, not available for plate reader-based screens or flow cytometry-based studies. And as such, we think that success is in part due to the image-based nature of this data. We can quality control this data very well, and we have this internal cytotoxicity readout. But we, of course, might be able to do better, and this is just a fraction of the research going on in the lab. But one thing we said is that so far we've just been using immunofluorescence and marker expression as a way to identify our target cell population, what might be cancer cells. As you see here in this image, the plasma cell markers in yellow and green identify plasma cells, and this is from a multiple myeloma patient, plasma cell cancer. So that defines in our classical workflow our target population. But we said there's so much more happening and there's so much tumor heterogeneity. Can't we use cell morphology to actually better identify cancer cells from non-cancer cells in this image, especially given that we have very large-scale data sets of billions of imaged and marker measured cells. So what we did is we trained a single convolutional neural network across some 300, 400 patient samples across several hundreds of thousands of weekly supervised or weekly annotated cells, just saying these were marker positive and these were marker negative. And we fed the neural network only our bright field and DAPI images rather than actually showing it the marker expression based on which we trained the neural network. And instead of looking at how well it performs, and there are many performance benefits compared to IF, the real killer performance evaluation that we can do with this kind of data is that we have matched for that particular patient, how for a subset of those patients, how well they responded to the treatments that we tested in our ex vivo assay. So instead of looking at performance or clustering of drug mode of action, we can actually evaluate the clinical response predictions of the neural network based on this post-hoc analysis of this data set. So this is our reference curve. This is how well the patients did on marker-based, immunofluorescence-based treatment recommendations that we just published. And you see that compared to prior treatment, this was the big improvement, and these are exceptional responders. But this scatter plot here shows immunofluorescence support for the treatment versus our deep morphology learning support. And you see that there's some, this isn't perfectly correlated, so there's some disagreement. We can look at how well the patients did whose treatments were recommended by our deep learning algorithm. And we see that they actually clinically do better than those that were following treatments recommended by immunofluorescence. And when we combine these two readouts, we get even better clinical response data. So This to us says that very likely DML, deep morphology learning, is further improving our treatment recommendations by considering only the morphology of the cells. And interestingly, there's also disagreement between these two. If we look at patients' responses for patients that receive treatments that were recommended by immunofluorescence, but not by the deep learning algorithm, we see that they actually do worse than just immunofluorescence alone. So our deep morphology learning really picked up some of the mistakes that immunofluorescence or marker-based cancer identification achieved in this data set. Now, a quick glance in what this neural network learned. We pulled out features, the activations of the last fully connected layer of the CNN that, again, just looks at Dapian bright field images. We call that the latent space. And then we can, cluster cells. For instance, tsne project the heterogeneity in that, or the structure of that data space for a few 100,000 cells. We see nice separation of cancer and healthy cells as the neural network was trained on, but there's also a lot of structure here. And I said in the beginning, we trained this neural network across hundreds of different patients, and they actually had different indications, different diseases. Now, what we see is that the neural network actually internally clustered typical morphologies for AML sample cells or CLL cells or DLBCL cells, both on the healthy cell side as well as on the cancer cell. And you can even turn it around and say, was therefore this clustering of the cell morphologies indicative of the indication of the disease that the patient had? And that indeed is the case. You get nice ROC curves diagnosing a patient based on this latent space morphotyping of these patient cells. So that's all from my side for now. Key collaborators for this were EXALT 1 and 2 trial, Medical University of Vienna, Philip Staber, Uli Jaeger, Julio Saperti-Furga at SEM, and Greg and Nicolas, who are now at Exentia, and Tim Heinemann from my lab for the label-free work. And I thank you for your attention, and I'm happy to take questions in the end of the question session or online for now.